the bond like H OH H OH. Now we draw the structure of adenosine. Adenosine, little wrong, no problem, it's okay, because we will see the detailed structures of the pyrimidine and purine in the later videos, so don't worry. Here we only understand the compounds and the enzymes, because component and enzymes are main focus of these chapters. So actually this is not like that. The bond is occur between this N and H. So this structure is called as citrulyl AMP intermediate. Now this citrulyl AMP intermediate reacts with aspartate. So as we know, aspartate is already formed inside the mitochondrial matrix, so it also come into the cytosol and reacts with the citrulyl. So the carbonyl group of citrulline and amino group of aspartate formed reacts with each other, and this reaction is a type of condensation reaction and form argininosuccinate. So argininosuccinate formed by the condensation reactions between citrulline and aspartate. So aspartate, which is already formed inside the mitochondrial matrix, reacts with citrulline, so AMP is released out and form a different compound that is called as argininosuccinate. So this is the structure of argininosuccinate. So aspartate reacts with citrulline, AMP is released out, and a different compound that is formed argininosuccinate. COO minus carboxyl group, CH2 CH, and there is COO minus another carboxyl group, NH amino, C N plus H2. Now NH CH2 3 CH, N plus H6 and COO minus, and this structure is called as argininosuccinate. Now this is here the enzyme that is this is 1 2 2, this is the enzyme 3. Now arginino with the help of what is the name of the enzyme? In this case argininosuccinate synthetase. Why argininosuccinate? So this is the enzyme number 3. And what is the name of the enzyme? Argininosuccinate synthetase. We later in this video we discuss about the enzymes, so don't worry about this. Now argininosuccinate form arginine and fumarate. So arginine and fumarate. So arginine and fumarate is formed from argininosuccinate. So this is the structure of arginine. H2NC, double bond N plus H2, NH, CH2 whole 3, CH, N plus H3 and C double O minus. So this is the structure of arginine. And what is the fate of arginine? Now arginine formed an important compound that is we want to get, that is urea and ornithine, and another compound is ornithine. So ornithine later enters inside the mitochondrial matrix from cytosol and initiate another round of urea cycle. On the other hand, urea, so urea is formed from arginine. So urea is formed from arginine. So what is the structure of urea? So C double bond O, NH2 and NH2. So in a single cycle 2 amino group is released. So this is the structure of urea. So in a single cycle 2 amino group is released, and that urea is formed from arginine with the help of an enzyme that is called arginase, and arginine and fumarate formed from argininosuccinate with the help of an enzyme argininosuccinase. So this is the whole process of the urea cycle. Now this is the enzyme 4 3 4. Here's the 4 and here's the 5. Now we understand the name of the enzyme. So first enzyme is carbamyl phosphate synthetase 1. So once again, carbamyl phosphate, carbamyl phosphate as the compound name, carbamyl phosphate synthetase 1. So name of the enzyme is carbamyl phosphate synthetase 1. There is another enzyme called as carbamyl phosphate synthetase 2, which regulates the important reaction, that is pyrimidine biosynthesis. On the other hand, carbamyl phosphate synthetase 1 catalyze the reactions of the formation of carbamyl phosphate in the case of urea cycle. Although the names are same, we can say that carbamyl phosphate synthetase 1 regulates the reactions of the urea cycle. On the other hand, carbamyl phosphate synthetase 2 controls the reaction of pyrimidine biosynthesis. Now what is the name of the enzyme number 2? So ornithine transform into the citrulline, ornithine accept the carbamyl group, carbamyl phosphate donates carbamyl group to ornithine. So the name of the enzyme is ornithine transcarbamylase. Ornithine transcarbamylase, So name of the enzyme ornithine transcarbamylase. So these reactions, number 2 reactions is catalyzed by ornithine transcarbamylase. Now enzyme 3, that is citrulline and aspartate form a compound that is argininosuccinate, and this reaction is also a ATP dependent reaction, and so the enzyme is a synthetase, and the name of the enzyme is argininosuccinate synthetase. So and the fourth enzyme, that is argininosuccinase, because argininosuccinate cleaves into arginine and fumarate enters into the TCA cycles. On the other hand, arginine continues the urea cycle. Now arginine cleaves into arginine, so this arginine cleaves into urea and ornithine with the help of an enzyme. So fifth enzyme is arginase. So these are the main 4 enzymes that are regulated in the urea, that regulates the whole urea cycle. So as you can see, this is the whole the process of the urea cycle. So first the glutamine, which is come from extrahepatic tissues, means outside from the liver cells. So the glutamine first enters into the livers, and glutamine transform into the glutamate and releases NH4 plus, and alpha ketoglutarate is formed. Now when the glutamate is transformed into the alpha ketoglutarate, it also releases NH4 plus and these ammonium ions, or you can say amino group, and so this ammonia with the help of a SCO3 form carbamoyl phosphate with the help of a 2 ATP. So this is the energy dependent step. Carbamoyl phosphate synthetase 1 regulates this reaction. After that, carbamoyl phosphate donates its carbamoyl group to the ornithine, so ornithine accept the carbamoyl group and form a citrulline. This reaction is the initial step of the urea cycle, and it occurs inside the mitochondrial matrix. Later citrulline enters into the cytosol of the hepatocyte and form a citrullyl AMP intermediate with the help of ATP, and aspartate, which is already formed inside the mitochondrial matrix, later enters into the cytosol of the hepatocyte, and amino group of aspartate and carbonyl group of citrulline reacts with each other and form argininosuccinate. It is the type of condensation reaction. Later argininosuccinate, and what is the name of the enzyme? The argininosuccinate synthetase. Why it is called synthetase? Because ATP is required in this phase. If the ATP is will not required in this phase, and in those cases it called as a argininosuccinate synthase. Now argininosuccinate cleaves into arginine and fumarate with the help of an enzyme argininosuccinase. So after fumarate enters into the TCA cycle, and arginine with the help of a enzyme arginase form urea and ornithine. As you can see from the structures of the urea, 2 amino groups are released, and this urea, after the formation of the urea, it enters into the bloodstream and travels to the kidney and finally excrete out from our body. So this is the whole process of the urea cycle. Thank you.